All aboard! Today, we're going to be talking about Grand Central Station in the cell, the Golgi apparatus. And we're off! The Golgi apparatus is another one of the organelles found in eukaryotic cells. It works in close conjunction with the endoplasmic reticulum, so if you haven't seen that video yet, now's a good time to check it out. But let's not get too far ahead of ourselves. So, Golgi apparatus. Well, that name's another real piece of work. But this time, it doesn't really mean anything in particular. It's just named after the guy who discovered it, Italian biologist Camillo Golgi. So far, so good. Now, what does this Golgi thing look like? The Golgi apparatus is essentially a system of flattened membrane discs called cisternae, all stacked on top of one another. The cisternae are those elongated yellow things you see in the picture right here. They're basically just membranes that are open on the inside. This space inside is also called a lumen. A stack of cisternae is called a dictyosome. And then a bunch of dictyosomes together forms the Golgi apparatus. A single cell might contain anywhere from one of these things to a lot of them. So from here on out, we're just going to be showing the Golgi apparatus as a single dictyosome. The Golgi apparatus also has a bunch of vesicles floating around it. Those are the little membrane-enclosed spheres you see here. Now, the Golgi apparatus can be divided up into its different functional areas. The first one is called the cis region. Cis is another Latin word meaning on the same side. The cis face of the Golgi apparatus is the side that faces, that is on the same side as, the ER or the nucleus of the cell. The cis face is the entry point of the Golgi apparatus. Now, if there's a same side, then there should also be an other side. And that'd be the trans region of the Golgi apparatus. This is the side that faces away from the nucleus, towards the cell membrane. It's the organelle's exit face. Between them is what's known as the medial region. The regions contain various enzymes, allowing them to take on a variety of different tasks. And speaking of tasks, so now we know what the Golgi apparatus looks like, but we still don't know what it actually does. The Golgi apparatus is basically Grand Central Station inside the cell. Imagine the proteins coming out of the ER as the passengers, and the vesicles that take them where they need to go are the trains. The trains take the passengers to the cis face of the station, where they get off the train. Basically what happens is that the vesicles fuse with the membrane of the Golgi apparatus, releasing the proteins inside. Once inside Grand Central Station, the passengers can upgrade their tickets. In other words, the proteins undergo certain modifications. For example, some proteins get sugars attached to them. That is, they undergo glycosylation. All this takes place in the medial region, with the vesicles transporting the proteins from one cisterna to the next. Then the passengers all get onto different trains headed towards their final stops. So the proteins get concentrated and packed into vesicles, which are sent off to different destinations throughout the cell. And all of this happens on the trans side, of course. Lipids also undergo further processing in the Golgi apparatus. The whole mechanism is similar to what happens with the proteins. Okay, so what happens to the vesicles after they leave the Golgi apparatus? They're taken to either the cell membrane or the lysosome. Once there, the vesicle membranes fuse with the other membranes. If the vesicles are carrying membrane components, these get immediately incorporated into the membrane. And if they're carrying secretory proteins, these get released out through the membrane. Alright, so that does it for the Golgi apparatus for now. But one more time, here are the most important parts. The Golgi apparatus is a cell organelle made up of a number of dictyosomes. The dictyosomes in turn are made up of cisternae. Proteins and lipids are brought to the cis face of the Golgi apparatus, modified in the medial region, and then leave through the trans phase where they're taken to their destinations. This takes place via transport vesicles. Alright, so now if you want to know what the lysosome does with the proteins from here, just keep clicking through the playlist.